Welcome to you, sir. Thank you. Be here, sir. Thank you, sir.
Thank you, sir. First of all, let me thank and appreciate Dr. Arnaba Malik, sir, Principal MLRIP, for improving the knowledge and skills of faculty through this type of faculty development programs. Actually, it is very much important. We have to improve the knowledge and skills of students, but also of course, the faculty also should improve or update themselves. Otherwise, we all teachers are outdated, and a teacher is a lifelong learner. So we have to learn the things from each and everyone. I thank Arnaba Malik sir and MLR College of Pharmacy Management for giving me this opportunity to interact with my fellow teachers. Shall I start the program, PPT, sir?
Yes, sir. 2 minutes, we'll have a small introduction, then we'll start the program.
Okay.
Yes, sir. Yeah, good morning, participants. Welcome to the fourth day national level 5-day faculty development program. So already we have completed 3 days, and we have seen a huge participation across the country for this faculty development program. So I hope today also we'll have the great session by our chief guest, Krishna Mohan, sir. So without much delay, we'll start today's session. So today's session will be handled by Dr. Vaishnavi, Assistant Professor, Department of Pharmacy Practice. Vaishnavi ma'am, you can start the session.
A very good morning to everyone. I'm Dr. Alambram Vaishnavi, Assistant Professor, Department of Pharmacy Practice, Mari Lakshman Reddy Institute of Pharmacy, Hyderabad. I take this privilege to welcome all of you to the fourth day of the Faculty Development Program organized by Department of Pharmacy Practice and Pharmacology, Mari Lakshman Reddy Institute of Pharmacy in association with APTI on integration of artificial intelligence and network pharmacy in modern pharmacological sciences. Today's session is on artificial intelligence powered genomic analysis, revolutionizing the detection of genetic mutations. I take this opportunity to introduce our speaker of today's session, Dr. Krishna Mohan Chinnala, Professor and Dean, School of Pharmacy, Nalla Education Society's group of institutions, Hyderabad. Sir has 25 years of vast experience in teaching and research, and sir has worked as a hospital and clinical pharmacist at Kingdom of Saudi Arabia, and sir is also a psychologist too. Dr. Krishna Mohan has completed undergraduation B.Pharm degree from Kakatiya University, Warangal, post-graduation M.Pharm degree from Andhra University, Visakhapatnam, and PhD from Kakatiya University, Warangal in pharmaceutical sciences. He has done his Master of Sciences in Psychology from Kakatiya University, Warangal. Sir has 115 publications in peer-reviewed, reputed national and international journals, and 60 presentation in national and international conferences. He has guided over 60 M.Pharm students, and 2 research scholars are pursuing their PhD under the sir's supervision. He is an editor, reviewer, and member of advisory board of several national and international reputed journals. Currently, sir is the chairman of Board of Studies Pharmacy at Nalla Narasimha Reddy Education Society's group of institutions, Hyderabad, and he has served as member of Board of Studies in Pharmaceutical Sciences, Kakatiya University, Warangal. Sir has unanimously elected as the president of APTI, Telangana from 2022 to 27. We are very proud of this. Dr. Krishna Mohan sir is a lifetime member of several professional organizations such as Indian Pharmaceutical Association, Indian Pharmacological Society, Association of Pharmaceutical Teachers of India, Indian Pharmacy Graduates Association, Laboratory Animal Scientists Association, Indian Toxicological Society, Indian Society of Technical Education, Association of Pharmacy Professionals, and Society of Pharmaceutical Education and Research. Sir has received many awards. Sir has received APP Distinguished Teacher Award in 2017 from Association of Pharmacy Professionals, Outstanding Scientist Award in 2022 from International Scientist Award on Engineering Sciences and Medicine. Sir has received the Best Pharmacy Teacher Award in 2022 from Telangana State Pharmacy Council, and Sir was been awarded as Most Socially Active Principal of the Year Award in 2023 from Telangana Information Technology Academy. I would like to request Dr. Krishna Mohan Chinnala garu to start the session. We welcome you, sir.
Thank you, ma'am. Thank you for your nice words about me and the introduction. Please confirm whether my presentation is visible.
Yes, sir. You're audible, sir. So you're visible, sir.
About presentation, ma'am, I shared the presentation.
Yes, sir. Your PPTs are visible, sir. You can take over the session.
Is it a full screen?
Sir, please put it in full screen, sir. It is not in full screen. Slide share, slide show, sir.
Okay, just wait for a while. Check it now, ma'am. Is it in full screen?
No.
Pardon?
F5, sir. Short.
Yeah, F5 copy everything. I am doing it, but I am unable to share it. Okay, I'll stop and I'll do it again. Just wait for a while. Bear with me. Share. Is it visible, ma'am? Just confirm.
Yes, sir. It is visible now, PPT.
Okay, but not full screen.
But just if possible, keep it in slideshow, sir. Otherwise, you can proceed, sir.
Okay, now? No.
No, sir.
Done. I'll continue this.
Yes, sir.
Okay.
You please continue, sir.
Sir,
Sorry
under
for
the
the
slide,
inconvenience.
Sir, under
Yeah.
the slide, there's something called 65%. Just go
Okay.
next. There's a button next to that on the left hand side. Just click on that, sir.
Which one?
Yeah,
65.
come to the right, sir. Little.
This one will increase the
No, no,
size.
here,
That's
here.
it, right?
Just there's something mentioned 65.
Okay.
Before that, there's a slideshow button. So just click on that, sir.
Yeah, I clicked it, sir. But in my laptop, it is showing full screen.
That one.
It is showing full screen in
Oh.
my laptop, but maybe
Some technical errors.
done.
Sir, okay, sir. You please continue, sir.
Yeah. Thank you. Without wasting of time of all eminent people. Again, good morning to all. It is my privilege to present in this faculty development program about revolutionizing the detection of genetic mutations. Okay, first we will discuss about a few things which may generate interest in the presentation. Okay, can anyone please confirm whether the slide is changed?
Sir, now the slide is showing like news from the Hindu slide. Third slide. You are in third slide.
Okay, right. So here, please check whether fourth one is like changed to fourth. Or
No, sir.
and then this one is not shared, I think. Okay, now is it the first slide now?
Yes, sir. It is in first slide now.
Second. I changed
Yes,
the
sir.
slide to second one.
Moving, sir. Yes, sir. Changing.
Yeah. Okay. Thank you. Thank you for the confirmation. So, as everyone said, I am working at Nalla Narsareddy Group of Institutions as a professor and dean from last 12 years at the School of Pharmacy. Ours is an integrated campus consisting of School of Pharmacy, School of Engineering, and School of Management Sciences. Recently, School of Pharmacy got even NBA accreditation also. Ours is an autonomous institution, of course, NAC A plus accredited. And we will go into our topic. I think everyone can see this slide. The news from the Hindu: woman becomes victim of clinical trials. Okay, so in a pharma company based at Hyderabad conducted clinical trials, and one lady named Chilki, she died. Okay, Chilkamma, her full name is Chilkamma from Mahbubabad area. So she died after clinical trials. Okay, so everyone blamed the pharma company that they have conducted unethical practices in clinical trials. That's why she died. Okay, but the response from Axis Pharma, who has conducted these clinical trials, woman's death not due to clinical tests. So who is correct then? So we have to check it. And as a pharmacy professional, we have to protect ourselves. No one will come to protect us, and everyone is ready to blame us, including the political leaders. See, in this news, the political leader Brinda Karath also blaming the clinical trials in India. Okay, based on these people's and few people's unethical practices, the even apex court, Supreme Court also issued a notice to center on illegal drug trials. And see, think about the clinical trials. Here we will discuss few things, right? About ethical conduct of clinical trials in India. Okay, so I'll give few examples here, which I read in news. Okay, see, this is long back news, but recently also around 30 BTech students from different from different colleges participated in a clinical trial. Okay, and the trial was 72 hours trial, and the company asked them to stay there at company premises only, and they have not allowed to use the mobiles. Okay, these all precautions to take to do a very good clinical trial. Okay, so and all of them are final year students, and they are not illiterates, and they have signed the written consent also before participating in it. And the scientist who conducted it explained everything about the drug and its side effects, its contraindications, its mechanism of action, everything. Okay, and taken written consent. And these 3 days they switched off the mobiles. And once they came out from the pharma company, they switched on, and the parents called them, and they informed the parent that we were in clinical trial. That's why we have not answered your calls and all. So the next day there was big, huge agitation against the company, saying that the company is using their wards as guinea pigs. Okay, so how far it is correct? And based on this type of news, the Enadu news people, they approached Professor Sarangapani sir, then principal of University College of Pharmaceutical Sciences, Kakati University, and asked him to explain about clinical trials. They want to like give every day a news on clinical trials. So sir asked them to sit and explain each and everything about clinical trials. At the end, what they said is, now we are educated about clinical trials. We won't publish any negative things about clinical trials in Inadu News. So I too observed at my home, I am getting Inadu regularly. So I to observe there is no negative clinical trials news in ENOD. So why I am saying this example is we have to protect ourselves, because see, one guy is attended clinical trial today here at Hyderabad and traveling overnight to Bangalore and attending in another clinical trial and getting 6,000 of rupees from the company. And who is the one going to be affected? Obviously, it is the drug manufacturer or the researcher only, not the candidate who is participating in. Okay, unless if it is having any drug interactions and all. Otherwise, the washout period is not completed for the drug, first drug, and is attending clinical trial at Bangalore with the second drug. So that's why pharma companies also like make some arrangements. Okay, like recruiting the volunteers based on their iris identification, right? Aadhaar and all iris identification. If the person is attending a clinical trial here at Hyderabad and moving to Bangalore and tries to attend, he or she can be like informed that already yesterday you attended one clinical trial, you are not eligible as a volunteer. Okay, so they are not allowing him. So such type of measures they are taking. Still, the one or two companies, if they are participating in illegal drug trials, and everybody is blamed by all the people, including the apex code. So what happened to this Chilkamma? That's what we have to discuss here in this session. Okay, so the fact for the death of Chilki is like many individuals believe that when they are prescribed a drug and it does not cure them of their illness, the doctor gave them the wrong prescription. However, in reality, the prescription is correct but is unable to take full effect due to the patient's metabolism. So that's why inter-individual variation came into the picture, precision medicine came into the picture, and personalized medicine came into the picture. So we have to think as a pharmacist, we have to think about the prescription drug and the full effect, whether it is there with the patient or not. That's why it is an important thing for conducting patient counseling regularly to all the patients. Okay, I'll give few examples here. If we get allergy, we will use levocetirizine. But if my wife takes levocetirizine, she will get allergy under her eyes and lips. Okay, so allergic reaction will be there. So it is made mandatory for me to give montelukast and fexofenadine combination. Okay, or montelukast and balestine combination, but not cetirizine or levocetirizine. And another example for my son, if he gets from the childhood, if he gets fever, paracetamol won't work for him. So in his childhood, what I did is I gave Nimslide. Okay, it it has so many side effects, but it was inevitable for me to reduce his fever. Okay, I tried increasing dose and all with paracetamol. Due to that, he got even seizures also in one instance. So I gave one dose, only one dose when he gets severe fever. Okay, it used to take care of his fever. Just because of his body nature or metabolism, he was unable to give full effect towards paracetamol. Okay, later the doctor advised me to give the paracetamol and mefenamic acid combination, but it also worked for, I can say, 75% only. Okay, so all these examples are giving the conclusion that the drugs are like working properly in a fixed or like a range of people only, maybe up to 60% population or responding to the drug. Then what about the remaining 40% patients? Okay, it is more important to know what sort of person has a disease than to know what sort of disease a person has, said by Hippocrates. Okay, so drug response, as I said, the patient group, okay, 60% of people are maybe responding to the drug, and remaining 40, in that 40, 10 may be reduced response. As teachers, we all know about subtherapeutic level of drug, which is pharmacologically no use at all, right? Pharmacologically inert, I can say, right? It is subtherapeutic, no use at all. And in few people, it is supratherapeutic, or it crosses the minimum toxic concentration, and it produces toxicity. So this and in another group of people, there will be no effect also. Okay, normal response and like cautionary response and toxic response, no response, altered response, all are just because of the metabolism of that particular individual. But the prescription is same with the therapeutic standard dose only. It is not increased dose or decreased dose, but different variations of effects we are seeing. Why it is happening? Okay, so just because of the genes, right? So what are genes? Genes are the biological units of heredity. Genes are specific sequences of bases that encode instructions on how to make proteins. We all know, we read in our pharmacology that drugs, most of the drugs, not all, most of the drugs act through the receptors, right? So receptors are nothing but proteins. We know that. But how proteins are synthesized? Just because of the like specific sequences from genetic material, okay, makes the proteins. If at all any flaw in genes can result a disease. Okay, normal physiology, pathophysiology, then pharmacology, then clinical pharmacy. This is a sequence we have to think about the treatment and everything. Okay, recently when I participated in Mallareddy College of Pharmacy or Mallareddy Vishwavidyapeeth, my teacher, Professor Rambo sir, said, if our students know pathophysiology and pharmacology well, they can be recruited as doctors in rural areas. Okay, that knowledge must be there with them to compete with doctors. Okay, always we are saying we are no way inferior to a doctor, but we have to impart the physiology knowledge, pathophysiology, pharmacology, then the management of disease and counseling part. Of course, they will learn on practice also, but these three parts in the sequence are important, right? So why I am insisting here is, if you know the physiology properly, abnormality in physiology is disease. Okay, so treating the disease, the classification of drugs and all is pharmacology, and patient counseling, how to manage the disease and everything will comes under clinical pharmacy and all, right? So genes carry the instructions that allow the cells to produce specific proteins such as enzymes also, right? We know few drugs acts through enzymes. The pattern of active and inactive genes in a cell and the resulting protein composition determine what kind of cell it is and what it can and cannot do. Okay, That's what the conclusion is. Flaws in genes can result in disease. Okay, how many genes does it take to make a body? And the complexity, based on complexity, the number of genes are there. And this slide is very much important. Please observe carefully. Okay, so DNA is information. Okay, DNA is our English language, and ATGC, okay, or your alphabets A, B, C, D. Then codon is a word, gene is a sentence, chromosome is a chapter, and genome is a book. Okay, always we are talking about genomic picture, means a book of your own genes. Okay, I'll read again. DNA is English, ATGC or your alphabets, and your codons are words, and gene is sentence, chromosome is a chapter, genome is a book. Right? So here genetic code deoxy ribonucleic acid contains all the information necessary to make a complete organism. So DNA is composed of a combination of 4 nucleotides. They are adenine, thymine, cytosine, and guanine. Just now we discussed ATCG. They are our alphabets in genomic picture. Okay, so the central dogma, of course, here DNA is the like proteins from RNA, RNA is from DNA. So a long double-stranded string of nucleotides that encode for many genes is DNA, and a single stranded copy of one gene is RNA. Then proteins are composed amino acids. Amino acids are made from triplets of nucleotides called codons, your ATGC. Okay, so here, if any change in the picture, in the genetic picture or gene, may lead to your change in protein structure. We all know that lock and key mechanism is applicable for our drug and receptor concept, right? So if the agonist, if drug is having structural similarity and affinity, may produce intrinsic activity. If structural similarity is not there, then the drug may not occupy the receptor. According to Professor S.S. Kulkarni, the eminent pharmacologist, 6,000 new receptors on our body are waiting for our research, for discovering a new molecule which can work on those receptors. Till now, no one detected or discovered a drug which can act on these 6,000 new receptors. Okay, so receptor shape here, if it is a box shape, the drug also should have the same to occupy the receptor first. Okay, then if it is a round one, okay, and these shapes are determined by your genetic picture, right? So it may alter your pharmacological action. Okay, so here a small change in gene sequence can result in a very different protein. Okay, see ATG. Okay, so if it is having just small difference in its codons, it may produce you different action. Okay, change in DNA are called as variations or mutations. This word we learned or heard about it during COVID. Okay, the COVID virus made so many mutations and got resistant. That's why so many people died and all. So this changes in DNA, whether it is in organism or human being, is called variations or mutations. Variations in genes may leads to full act, full effect of drug or less effect of drug or toxic effect of drug or altered effect of drug. Anything is possible because of alteration in your genes. variations in DNA genotype can cause observable changes in individuals. Okay, variations in our DNA make us unique. See the picture, all faces are not similar. Why? Because the variations in the genetic picture. If not, this will happen. Entire family will have the same face and all. Is it possible to get this type of faces for entire family? Actually, it is not possible. It is actually uh, created by an app, that is Dub Smash app. You can also create such type of picture. Okay, so here this type of if any no changes at all in gene picture, you can expect this type of picture, right? So human genetic variations in DNA, single nucleotide polymorphisms, they are called SNPs. Okay, so insertions, deletions, short tandem repeats, and translocations, everything is possible in variations. As this conference or faculty development program is of pharmacology, I can give few examples here for insertion and deletions. Okay, so insertion of your genes and getting a modified altered or modified end product. The best example, National Institute of Nutrition, Tarnaka, Hyderabad, created obese rats. How they created? They have inserted obesity genes into the rats and got that obese rats, and they are very much useful for your obesity studies and anti-hypertensive, anti-diabetic research. Okay, so this is possible. I want to say that one. And deletions, of course, previously what we did for anti-cancer activities, we have induced cancer by injecting cancerous cells into mice. Okay, and used the Vernier calipers to measure the growth or size of tumor. Okay, the main obstacle while measuring the uh, tumor with the digital Vernier calipers is its hair. So what scientists did? They have removed the gene which is causing production of hair follicles and produced nude mice. Now see, nude mice are very much useful in cancer research. So such type of deletions and insertions is possible to make genetic variation, right? So what are SNPs? Okay, a single nucleotide polymorphism is a small genetic change or variation that can occur within a person's DNA sequence. Okay, one letter variation in DNA sequence and SNPs contribute to a difference, contribute to differences among individuals. That's what we discussed. Why individualization or individualized treatment is important, or personalized treatment is important. See, here in SNP, just one change is there. Instead of T here replaced by C, and it will give a different variant of your gene, right? So the same here, instead of CG here, TA is a, it, and what is happening if the change is what is the net result with the change? So common sequence is like this, and variations. Okay, here the polymorphism occurred because of the polymorphism, the DNA variations are taken place in that particular person. And already we discussed about deletions and insertions and translocations. Okay, all are possible in polymorphism, and This slide again important for every pharmacologist, and DNA sequence variation in a gene can change the protein produced by the genetic code. Already we discussed about the lock and key mechanism. So this gene is producing the protein product with this structure. Okay. And second, next variation in genes is producing a different type of structure of protein, or it is nothing but your receptor. Then how can a drug will sit and elicit the pharmacological action? Okay, here already we discussed not only the suitable key may enter into the lock. After entering into the lock, the lever should be adjusted or suitable to the lock to open. Otherwise, it may not be opened. Okay, structural similarity is there with the drug, but it is not producing any action because of its lack of intrinsic activity. So it is mandatory to have structural similarity first to become an agonist or even an antagonist also. Then it depends whether it is producing pharmacological action or not. It depends agonist on the antagonist, right? History of this gene changes and all. Okay, we already studied in BFOM itself about N-style transferase gene. Okay, divides the people into slow stylators and rapid stylators by taking the example of Caucasians or like the people who are a few people are not tolerable to N-style transferase also. Okay, such type of examples are giving that the individual genes are altering the drug action. Okay, so then what is pharmacogenetics? Is generally regarded as a study or clinical testing of genetic variation that gives rise to differing response to drugs. Okay, pharmacogenetics refers to genetic differences in metabolic pathways which can affect individual responses to drugs, both in terms of therapeutic effect as well as adverse effects. Pharmacogenetics, how relevant are your genes to drug response is nothing but your pharmacogenetics. Pharmacogenomics, pharmacology plus genomics is called pharmacogenomics, right? So factors contributing to inter-individual variability in drug disposition and action. So all these we studied in pharmacokinetics. Age, race, weight, gender, concomitant diseases, concomitant drugs, social factors. Last but not least, and important one is genetics. So these are main contributions for your personalized medicine. We have to consider all these factors, including genetics, while thinking about personalized medicine. Okay, what are the reasons a person would react differently to drugs is important to know. Okay, having a receptor, drug and receptor is like a receptor is a protein, drug is a ligand. Okay, what it is doing, it is sitting on the receptor and eliciting pharmacological action. Of course, a few may interfere with the intracellular genetic picture and alters the action, few may not, but it is important that the structure of the receptor is similar to structure of the ligand or the drug. And why variation is there? So here, if the drug structure and receptor structure is similar, uh, it may occupy receptor and elicit the pharmacological action. And here hypersensitive action also possible with more receptors present on the like surface of the cell or so, right? So next is hyposensitive, less number of receptors And next is there is no response at all. There is no structural similarity at all with the drug or ligand. So these things may alter your pharmacological action, right? Okay, already we discussed about lock and key mechanism. Of course, structural similarity and the structure of livers is important in drugs. Nothing but your side chains of a drug, pharmacophore or the molecule, main molecule, is important to act on the drug effectively. Okay, other physiological traits are there. Hypersensitivity, we discussed about it, and this may be useful in inter individual variation of drugs, and we can decide whether the tumor side acting on the receptors or not. Okay. HER2 receptors, okay, which are involved in breast cancer of women. Okay, if they are responsive towards the tumoroid given by the doctor, then only you can continue the drug. Otherwise, you have to discontinue it and you have to like give other drug. Okay, why? Because cancerous patients have less time. Okay, you have to give quality of life for them, improve the lifespan. For that, no trial and error method should be followed, like what the doctors will do for antibiotics. They will give for 3 or 4 days and ask the patient to come back and they will change the drug again. Okay, not like that. It is a life and death issue. So you have to think about how to give a perfect medicine to a cancerous patient to treat tumorous side. Okay. So for that, you have to check with the whether the cells are responding towards the drug or not with the biopsy of the breast cancer cells, right? So phenyl thiocarbamide is an example. So here is a story. What happened? This one scientist in the lab, while transferring this phenyl thiocarbamide into another container, it fall on his tongue and the adjacent scientist tongue also. So the adjacent scientist made a complaint that it is so bitter in taste. But the person who was transferring doesn't feel that it is bitter in taste. So then this genetic variation and drug action came into the picture. Okay, this is used even for paternity tests nowadays as a basic reference test. Okay, Mr. Shankal Dayal Sharma son claimed that Shankal Dayal Sharma was his father. Right? It is proved by this test only, paternity test. It was proved. Right? So here, this is the basic thing to know how the genetic variation, like giving effects, even simple taste also, bitter in taste and no taste at all. So think about the drug action now. Okay, how your body processes the drugs after receiving it is also important, and that is called your ADME, your pharmacokinetics, absorption, distribution, metabolism, and excretion. Of course, all of us know that metabolism and excretion together called elimination. So here, this is again important for our topic, metabolic enzymes. So enzymes which are metabolizing the xenobiotics or food, whatever it may be, are present in our liver. Okay, so they are called cytochrome P450 enzymes, DNA variations in special proteins in the liver called enzymes can influence a person's ability to metabolize certain drugs. Okay, DNA variations in special proteins called P450 enzymes, cytochrome P450, CYP450 enzymes can influence a person's ability to metabolize certain drugs. We all know if the drug is optimized manner metabolized, it will give optimized effect. If the metabolism is slow, it may leads to accumulation and toxicity. And metabolism is rapid, it may enter into subtherapeutic level only. It won't reach the therapeutic window or your margin of safety or whatever it may be. Okay, it won't produce any pharmacological action because of rapid metabolism. It won't reach the plasma concentration in optimized manner, right? The role of genetics in drug metabolism, 20 to 95% of individual variability is genetic based. Okay, it is now clear that virtually every pathway of drug metabolism, transport, and action is susceptible to genetic variation. Within the top 200 selling prescription drugs, 59% of the 27 most frequently cited in adverse drug reaction studies are metabolized by at least one enzyme known to have genetic variations, gene variants that code for reduced or non-functional proteins. So many other factors such as already we discussed, age, physiological functions and all. But the main factor here is gene variants of cytochrome P450 enzymes. So consequences of polymorphisms, what are the consequences we can expect or going to happen? May result in a different amino acid or stop codon, may result in a change in protein function or quantity. Okay, already we discussed about the importance of quantity also, function and quantity. If less quantity is there and maybe subtherapeutic level and more quantity of the drug and more receptors are there, you may get hypersensitive reaction also, right? May alter stability of mRNA, then no consequence also happen, no action at all from the drug. And what are SIPs? As part of the like inborn system for clearing the body of xenobiotics, the cytochrome P450 oxidases are heavily involved in drug metabolism, and genetic variation shifts affect the large populations. Inter-individual variability in drug response is just due to genetic variation in CYP enzymes. One member of the CYP superfamily, CYP2D6, now has over 75 known variations. See, 75 known variations means it will alter the effect of drug in 75 ways. Okay, we don't know the net result or the consequence, already we discussed. Okay, some of which lead to no activity and some to enhanced activity. And as estimated, 29% of people in parts of East Africa may have multiple copies of gene and will therefore not be adequately treated with standard doses of drugs such as painkiller codeine, which is activated by the enzyme and gives the toxic effect for them. Okay, after metabolism, right? So, and always we are discussing as a pharmacologist that every drug is having side effects, and all side effects may not be adverse in nature. Adverse in nature. So, for example, chlorpheniramine maleate tablet is there, CPM, or the popular Avil tablet. So, if you approach a doctor with MD physician with insomnia, what he prescribes is placebo, a placebo which is inert pharmacologically, or else he may prescribe Avil tablet. The side effect is used here. Okay, for therapeutic purpose, side effect is all of us know the drowsiness. Okay, So if it is RMP doctor, he may prescribe you the sleeping pill. The major problem with sleeping pills is addiction liability. So, but physician will think about your status of insomnia, may give you placebo or a will tablet to give you the relief from insomnia. Okay, why I am giving this example is we are using the side effect of drowsiness for positive purpose in treatment. The same way, the metabolism, you should not blame the poor metabolizers and say that they are not at all useful. Not in all times, not in all times. Poor metabolizers absent or greatly reduced ability to clear or activate drugs. Already we discussed, if poor metabolizer is there, the drug may not reach the uh, plasma into therapeutic window. Okay, maybe enters into plasma, but subtherapeutic level. Intermediate metabolizer, heterozygotes for normal and reduced activity genes. Okay, then normal metabolizers. It is normal and it lays between minimum effective concentration and minimum toxic concentration in the therapeutic window and gives the normal effect. And ultra metabolizers already we discussed. The ultimate concentration of drug may be reduced for pharmacological action. Okay, greatly increased activity, accelerating clearance or activation. Okay, here I'll give an example which where you poor metabolizers are useful therapeutically. Proton pump inhibitors are used to treat acid reflux and stomach ulcers. Ulcer cure rates using omeprazole and amoxicillin by CYP2C19 phenotype is like this. Rapid metabolizers, it is 28.6% only. Intermediate or you can say normal metabolizers, 60% only. And poor metabolizers is 100%. So think about a genetic variation also here with this example. Okay, intermediate metabolizer 60%. Maybe whatever the drug we are giving, those 60% have intermediate metabolizers, and those remaining 40% may have rapid metabolizers or poor metabolizers. Okay, it may be useful or harmful, we don't know. So importance of pharmacogenomics, one size fits all drugs only work for about 60% population, only 60% population at the best. And the other 40% of the population increase their risk of adverse drug reaction because their genes do not do what is intended of them. Okay, is it clear? One size fits all drugs only works for about 60% of the population at best. Okay, and the other 40% of population increase their risk of adverse drug reactions because their genes do not do what is intended of them. And what is the novelty in it then? Drugs of future, amazing new medicines will be based on DNA. Find out how they will change your life. This is what the novelty with the pharmacogenomics. Of course, here normal genes, okay, normal variant, normal action. Normal gene, normal variant, normal action, and SNP variant gene and abnormal activity. And if you want to make some changes in the gene and produce suitable receptor or a drug, okay, then altered action will be there. Maybe 100% successful treatment is possible. Okay. So the right drug for cancer, pharmacogenomics is used in targeted therapy for cancer to identify the best drug regimen for a particular tumor. Even tumors of the same type, such as lung, breast, or liver, vary at the genetic level. Okay, not all the cancerous cells are same in like breast cancer, lung cancer, or liver cancer. Okay, cancer is fundamentally a genetic disease, but most of the genetic differences between cancerous cells and normal cells are not inherited, they accumulate as the cancer develops. Okay, that is a major problem with the treatment of cancer. The drug is unable to identify the normal cells or cancerous cells. Okay, that's why they are affecting highly proliferative cells like bone marrow cells and all, including cancerous cells. Okay, analyzing specific genes in a patient's tumor helps doctor identify the drug combination to which the tumor will most likely respond. For example, the breast cancer drug Herceptin is only effective when the tumor cells have accumulated extra copies of HER2 gene and have high levels of the protein this gene encodes on their surfaces. So we will discuss about this with the pictures, okay, but the drugs which are developed by personal like pharmacogenomics mechanism or like this. So targeted therapies, Herceptin already we discussed, which is used in breast cancer for women. Okay, so this is useful in treatment of HER2 positive metastatic breast cancer. And Gleevec is the drug treatment for patients with Philadelphia chromosome positive chronic myeloid leukemia. And erlotinib treatment for non-small cell lung cancer, most effective in epidermal growth factor receptor positive tumors. And Maraviroc treatment for HIV, studies have incorporated a screening process for different receptors that HIV uses to gain access to cells. And iloperidone used for schizophrenia treatment. Company identified a genetic marker that predicts a good response to the drug. Okay, personalized drugs, Herceptin for breast cancer, Erbitax, okay, colorectal cancer, Tarceva, lung cancer, okay, Statera, then attention deficit disorder, 6-mercaptopurine, leukemia, metabolism, then antivirals resistant based on form of HIV, etc. And the list is growing rapidly by using pharmacogenomics. The scientists are discovering new molecules which can act through receptors and which can cure the cancer effectively. So for breast cancer in 1990, surgery was the first option, then radiation, then chemotherapy. Okay. Then in 2012, surgery, radiation, chemotherapy. Now it is specialized treatments for certain types of breast cancer. So specialized treatment will give like early detection, early treatment to the patient and may cure entire cancer or extends the life of thus that cancerous patient effectively. Here is the best example with Herceptin transgumab, used in metastatic breast cancer, targets tumor cells that overexpress the human epidermal growth factor receptor 2, HER2 receptors. Okay, best response attained in women who overexpress the HER2 receptor. HER overexpression in breast cancer cells should be done before patients receive the drug. That's what I'm saying. You should not waste time in on trial and error method. So here what you have to do, you have to take the biopsy, okay, breast tissue biopsy, and check with Herceptin activity. Okay, if Herceptin is giving positive response, the HER2 is giving positive response towards Herceptin, without any hesitation, you can start the treatment. And if it is giving negative response, then you have to give or try on biopsy tissues with other drug other than Herceptin, okay, to check whether it is giving positive response or not. HER2 gene product is overexpressed in 1/3 of breast cancers. And if it is overexpressed, sensitive to herceptin, you can give the drug. And here is a like screening test for HER2 slides, how it appears, and positive, it treats the cells. Okay, but negative, it won't. The cancerous cells will be, the multiplication will be same or increase. So P-glycoprotein as a novel target for drugs, P-glycoprotein like is a well-characterized human ABC, ATP binding cassette transporter of the MDR, right? So what it is doing is it is stopping the efflux of drugs. The major problem with the cancer treatment is the drug is entering into the cell, but due to the efflux procedure of that cell, the drug is unable to retain in the cell to show its full effect. That's why it is coming out. Okay, P-glycoprotein functions as a drug efflux pump. So with genomic changes and all in the receptor with the P-glycoprotein, we can stop that drug efflux. See in this slide, so the drug entered into the, in first one, drug entered and efflux was taken place and drug is coming out to cellularly. And there is no use or less use of anti-cancer drug. But in pharmacogenomics, what we are doing is we are blocking this efflux. So effectively, total entire drug will retain in the cell and kills the cancerous cell. And of course, if it works, you can reduce the dose also to kill the virus. You kill the cancerous cell instead of giving high dose and expecting adverse drug events and all. So you can achieve this with the pharmacogenomic technology. Multi-drug resistant inhibitors can block the pre-glycoprotein of resistant tumor cells and restore sensitivity to cancerous cells, cancerous drugs, right? So gefitinib is also a new product which is there in the market. And now we will discuss about the benefits of pharmacogenomics. Okay, creation of more powerful drugs, therapies more targeted to specific diseases, better, safer drugs the first time. No need to do more and more trials and waste money and time. Okay, it eliminates trial and error methods for finding optimized drug. Okay, more accurate methods for determining appropriate drug dosages. Just now we discussed about dose increasing, decreasing, or slow escalators, rapid escalators. After knowing the genetic picture, if it is slow escalators, you can reduce the dose. If it is rapid escalator, you can increase the dose to get therapeutic effect or to reach the drug into your therapeutic window and gives effective pharmacological action, right? So advanced screening for diseases to finding out the receptors and all also we can do it. And better vaccines will activate the immune system but will be unable to cause infections. Okay, so improvements in the drug discovery and approval process, easier development of drugs using genome targets. This is what important for the research scholars here. And decrease in the overall cost of healthcare. Obviously, whatever the treatment you are thinking, or the government also will think about affordability of medicines. Okay, so you think about cheap medicines which can reach to all the people. That's why India is popular to supply the medicines for less cost when compared with the developed countries to almost all countries, right? So even during COVID also, we saved the lives of so many people across the globe, not only India, because so many vaccines developed by Western countries, the cost was high. So the countries which were which are developing countries or underdeveloped countries procured vaccine from India and saved the lives of their citizens. So it is very much important about the cost also, along with effectiveness. Decrease in the overall cost of healthcare is top priority for the governments to give effective medicines to the people to reach. And prevention is better than cure always. And 4 lakh wrong drug prescriptions per annum are reported. Here, I can give a joke. There was a conference in US. Every country representative was giving their statistics about the adverse drug reaction taken place in their own country. When the turn of India, the representative went there onto the stage and said zero adverse drug reactions in India. Everybody was shocked. Okay, so much of population, 130 crores of population, and no adverse drug reactions. Then the representative, the scientist said, there are no statistics at all in India. So that is the reason Indian Pharmacopoeial Commission started taking care of reporting of adverse drug reaction. My request to all pharmacologists here, please involve in it. Please teach your students how to report adverse drug reaction to Indian Pharmacopoeial Commission or PVPI program, Pharmacovigilance Program of India and all. And if more adverse drug events are identified from your college or your end, especially from the colleges who are having formed, may establish AMC, Adverse Drug Reaction Monitoring Center, with the permission of IPC. You can apply for that. I hope few colleges in Hyderabad already got AMC and doing excellent job in identifying or reporting adverse drug reactions, and encourage the students to participate in pharmacovigilance program which are conducted by IPC also, so that we can encourage the citizens, the pharmacists, the doctors to report adverse drug reactions, and then we will get exact statistics of adverse drug events in India, right? And patient benefits from pharmacogenomics is maximize treatment success by individualizing patient treatment to match their unique genetic makeup. So here we can discuss about the doctor's practice. If so many times we will use that word, this doctor is good for the treatment of diabetes. This doctor's just if you visit his chamber itself, half of the disease will be cured. Why? Because it may be his uh, hospitality, how he is treating you, how he is the way he is talking to you, and at the end, obviously, the treatment also. Okay, if the treatment is good and just you are visiting for the a symptomatic relief, you got it in 1 day or just 2 days or 3 days, like in a little span of time, then you won't shift to other doctor. You will continue with the same doctor for you for entire your life. That's what is important even for the doctor also. So that is possible here, like one drug suitable for all is a wrong concept, and trial and error method, whatever the doctors are following may be avoided, and they may give 100% treatment here. Okay, so opportunity to build the practice also and keep current with the latest advances in genetic science. And patient benefits already we discussed with the cheaper medicines may be available for the patient, and no need to repeatedly approach the doctor or go to hospital, and duration, the dose frequency, everything can be reduced. okay, based on his disease and treatment, and provides the personalized and clinically relevant drug or drug gene interaction information. Helps the patients, okay, physician optimize the safety and efficacy of prescription regimens and proactive treatment choices. And Genelex DNA drug reaction test. So now there is a question, how to check my body genome picture, or at least the CYP2D6 metabolism pattern. Okay, so there are so many like in research works were conducted on it, and now the instruments also available in the market to check the metabolism or polymorphisms of CYP2D6 or CYP enzymes of your own body, and the literature is available if the enzymes are metabolized by CYP2D6 in this pattern, whether the drug is useful or not, whether the drug will give full effect or less effect or toxic effect. So with that, you can decide that, or the doctor and you can decide to use the medicine or discard the medicine. That beauty is there. Now, Genelex DNA drug reaction tests currently offer straightforward genetic tests that reliably identify and classify for the highly polymorphic cytochromes CYP2D6, CYP2C9, CYP2C19, CYP1A2, and Na2 into their slow, normal, and ultra-fast metabolizing forms. Okay, these enzymes process half of the most commonly prescribed medicines, including many with narrow therapeutic indices. And here we have to discuss an important thing. If a drug is having narrow therapeutic index, okay, doctor monitoring is mandatory, like digoxin. Okay, digoxin has a narrow margin of safety. If you increase the dose in micrograms, it may crosses the minimum toxic dose and enters into toxic area, produces toxicity. And widest margin of safety, the penicillin is having widest, but it is not there in the market. The main reason for that is 1.5 grams of drug you can inject intramuscularly into the body. That is the beauty of penicillin. But the major issue with the penicillin is hypersensitivity reaction. Again, the different genes involved here in hypersensitivity reaction. That's why it is not there in the market. Otherwise, it is a very good antibiotic with broad spectrum activity. And see, the margin of safety is wider. So you can inject even 1.5 grams intramuscularly. So that is what we have to think about narrow therapeutic indices drugs or wide and hypersensitive drugs also. Because here, just because of genetic picture, sulfa drugs and penicillin is not that widely used nowadays. Okay, so think about the drugs and genes. Half of the patients have genetic variations in these genes that lead to altered or absent function, resulting in elevated patient susceptibility to adverse drug reactions. Genotyping avoids adverse drug reactions. Okay, so think about your genomic picture and how it is producing adverse events in normal treatment, and check with your polymorphic status of cytochrome P450 enzymes in your body, and then check with the drug. Okay, then you won't get adverse drug reactions and you will get 100% treatment for that disease. Okay, Roche AmpliChip 450 test. This is the instrument available in the market. Okay, test is intended to identify a patient's CYP2D6 and CYP2C19 genotype from genomic DNA extracted from a whole blood sample. And what I heard is nowadays no need to give us blood sample also. With your saliva also you can check this type of test. So that is the advanced technology introduced in the market also, right? So barriers for pharmacogenomic process, limited drug alternatives are available, like transjumab we discussed. Okay, even though it is not producing 100% efficacy, if it is producing 60 or 70%, if any alternative is not there for the drug, it is inevitable to use the transjumab in like breast cancer in women. Okay, that is what the barrier for pharmacogenomic progress, right? The complexity of finding gene variations that affect drug response. So still, people are not like familiar with finding out their gene variations and all. So disincentives for drug companies to make multiple pharmacogenomic products. So no company is coming forward to do this type of research. Of course, most of the companies thinking about the profits only, not the exact research and benefit to individuals. I'm talking about individuals. But few companies are conducting this type of research, and the best example is Melinda Gates Foundation is giving or supporting this type of research to give inter individual treatment for rare diseases or cancer disease. Okay. Scope for the pharmacogenomic, the emerging discipline of pharmacogenomics attempts to apply the innovative technologies of genome sequencing in order to better understand drug response, to produce more effective drugs with fewer side effects on the basis of individual patients' genetic makeup, and making personalized medicine an economically viable possibility. All we have discussed about it. And pharmacogenomics and the future, it is more important to know what sort of person has a disease than to know what sort of disease a person has. Initially, we discussed about it. And pharmacogenomics, though in its infancy stage, endeavors to tailor medical care to the individual. Okay, the goal of personalized medicine, this already we are using everywhere as a pharmacology teacher or pharmacologist. The right dose of the right drug for the right indication for the right patient at the right time. All rights must be reached to the patient, then your treatment is right. Okay. In near future, like if you go to any like mall, you are selecting the like your clothes or dress according to the generalized size, like small, medium, large, XL, triple XL, double XL, and all, right? Or else 40, 42, 46, 36, etc. But it may not be 100% suitable for your body structure. But if you go and approach a tailor, he will take the measurements of your body structure and stitches a dress which is 100% suitable for your dress. Okay, why I'm giving this example is you may do that with the drugs also. In the future, we have the ability to tailor drugs according to each individual's specific genetic makeup. That indicates there is no flaws in it and you will get 100% treatment. That's what I'm saying. 100% suitable dress you will get, not loose, not tied. Okay, the same thing here. With the treatment also, tailoring of medicines, you will get the suitable medicine for your treatment. And traditional method was, okay, the patient gets sick, patient goes to a doctor, okay, doctor checks the symptoms and write the prescription, the patient uses the prescription. If it is cured, he won't come to the clinic again. If not, he will come back again. Today's method, patient gets sick, patient goes to doctor, checks for symptoms, and doctor asked for diagnostics tests like blood analysis or urine analysis or blah blah blah, all. Okay, and doctor determines the problem or the cause for the sickness. Then he will prescribe the drug. Here also, maybe 60% or 70% people may get relief from his disease. And what about remaining 30 to 40% people? They may not get the relief from clinical symptoms. So, what is the uh, scope or what is the future for this problem, solution for this problem, right? So patient sick and comes to doctor, doctor checks the symptoms and asks the patients to bring genetic picture of their own. Okay, that particular patient genetic picture he will ask. Based on the genetic picture, the doctor will analyze the symptoms, analyze the disease, and gives the treatment, which may give 100% treatment, and the patient may not come back to the hospital again. This is what possible with the pharmacogenomics or precision medicine or individualized medicine. Okay, pharmacogenomics is determining the right drug in the right dosage at the right time for each and every patient, right? So about DNA in April 1953, okay, Watson and Crick determined the structure of DNA. But in 2003, Human Genome Picture Project was determined. Okay, in April 2050, you wake up feeling terrible and you know it's time to see a doctor. In the office, the physician looks your order, okay, listens to your symptoms and decides to prescribe you a drug. But first, the doctor takes a look at your genome picture. Okay, today's versus future. Today, drugs are one size fits all, and future drugs specific for you. Okay, this may be possible in 2050. You may have in your wallet this type of card, like your credit card, debit card, or like whatever may be, like with QR code and all, which contains your entire genomic picture. Okay, it may be possible in future, right? So here what the patient is doing, she is handovering the card to the pharmacist. This doesn't mean that self-medication can be possible. It is dangerous. No, doctor is doctor, pharmacist is pharmacist. A pharmacist will check the symptoms. Of course, if it is simple disease can be treated with OTC drugs and he will give the OTC. Otherwise, he will advise the patient to meet the doctor to get the prescription. Okay, so if you have thorough knowledge of pharmacogenomics as a pharmacist, you can impart the knowledge to the doctor and patient. You can work as a connecting link between these two people, because the doctors may not have that much information about pharmacogenomics, and patient also, right? You are the one to take care of these pharmacogenomic picture of the patient and inform the doctor too, right? If any drug interaction is there after prescription also, with your pharmacogenomic knowledge, you have to inform the doctor to change the prescription, right? So here most of them are Telugu audience, so that's why I kept this slide. Okay, మీరు కారు శరీరం కారు లాంటిదే, కియా టాటా ఫోర్డ్స్ ఇలా ఒక్కో బ్రాండ్కి ఒక్కో డిజైన్ ఉంటుంది. ఇది జెన్యు లాంటిది, తయారీలో ఒక్కో రకమైన మెటీరియల్ను వాడతారు. స్పేర్ పార్ట్స్ను మనిషి శరీర భాగాలతో పోల్చుకోవచ్చు. బలహీనమైన వ్యవస్థ వల్ల రోగనిరోధక శక్తి తగ్గుతుంది, తరచూ ఆసుపత్రి పాలవుతారు. నాసిరకం పరికరాలతో బండి పనితీరు దెబ్బతింటుంది, సగం రోజులు గ్యారేజ్లోనే ఉంటుంది. జన్యు సమస్యలు ఆయువుని మింగేసినట్టే, డిజైనింగ్ లోపాలు వాహనం జీవిత కాలాన్ని ప్రభావితం చేస్తాయి. వాహనానికి ఇంధనం ఉండాలి, మనిషికి ఆహారం కావాలి. బండికి సర్వీసింగ్ లాంటిది, మనిషికి ఆరోగ్య పరీక్ష. తేడా వస్తే బండి బ్రేక్ డౌన్ అవుతుంది, మనిషి ఐసీయూ పాలవుతాడు. సో యూ ప్లీజ్ చెక్ యువర్ genetic picture, okay, and think about genes and all, right? So it is very much important to think about it. And now at the end, the conclusion remarks are AI powered genomic analysis, the revolutionizing detection of genetic mutations. Till now we discussed about the basics, how genetic mutations occurs and how to do detect it and what are the instruments used, and basic things. Okay, so here artificial intelligence can be used in everywhere in pharmaceutical research, right? So here genomic analysis involves studying of DNA sequences to identify variations and mutations. Already we discussed this. And traditional methods are time consuming and require extensive manual interpretation. That's where AI is involving and helping us. Like HPLC previously, what we did, we used to take the samples and inject into the column and turn on the pump and everything. But automated now, everything it will take care of and give you a printout of the result. Okay, such type of automated or advanced technologies, or whether it is AI or other technologies, can be used to improve the efficacy and the safety also of drugs. Okay, so integration of AI has transformed speed, accuracy, and scalability. Role of artificial intelligence in genomics, AI, especially machine learning and deep learning, can process vast genomic data sets efficiently. In our previous slides, in the disincentive sign, what we discussed is unavailability of gene variations. Because of this AI tools, we can analyze them in a rapid manner and give the genomic picture to the patient. Okay, and a doctor also will learn the things with the AI, how to identify, how to detect the clinical symptoms of that genomic picture and the cause for those like pathophysiology symptoms. Then he can prescribe the medicine. Okay, identifies the pattern in DNA sequences that are difficult for humans to detect. This is also another advantage for that, and learns from existing genomic databases to improve prediction accuracy. Obviously, we need accurate results. Otherwise, if your gene picture is not accurate, and you may not get effective treatment. Okay. Types of genetic mutations detect single nucleotide polymorphisms, insertions, deletions, copy, number of variations, and structural variations. All we already discussed. It is possible with AI. That's what I want to say here. Advantages of AI-powered genomic analysis: high speed, rapid analysis of whole genomic sequences is possible. Improved accuracy, reduces human error in mutation detection. Scalability, handles large-scale population genomic studies. Here it is very much important, as all pharmacy teachers, we know population pharmacokinetics. Okay, this scalability is important in POP-PK. Okay, early diagnosis enables early detection of genetic disorders, may give you the best treatment here. I'll give an example of my junior. When we were in B Form, our physiology teacher used to note down the blood group in the register itself after conducting blood group experiment in physiology lab. And one of our junior, Mr. Srikant, okay, his WBC was abnormal, and the teacher, Professor V. Venkateshwar sir advised him to like check it outside also. Maybe experimental error is there, but he ignored it, and with the curiosity, interest, and by force from parents, he appeared for MSET again and got dental BDS in Siddhartha College then, and joined there. And the same experiment was he performed there also. It was abnormal. It was abnormal, and the professor insisted him to bring the report the next day. So, and he checked with the diagnostic center. It was abnormal, but at that time it was already late. He was suffering with leukemia. You believe it or not, the next year he died. So such type of small examples, please, all the teachers, please take care of conduct the experiments ethically and check the reports and advise them. Of course, if that guy would have done what my teacher said or followed his instructions, he would have alive. So detection of uh, diseases, whether it is genetic or cancer-like diseases, may save the life or may extend the life. That's what I want to say here. And cost efficiency, already we discussed a lot about it. Okay. And key AI techniques used is machine learning, deep learning. Okay. Then natural language processing for genomic literature mining. Already we discussed about the dictionary and all. Okay, ATGC, etc. Okay, predictive analytics for mutation impact assessment and applications in healthcare. Personalized medicine is possible with AI. Cancer genomics is possible, and already we discussed about rare disease diagnosis and treatment also. And of course, drug development, drug identification, the target identification and pharmacogenomics, both are possible. You may change, you make changes in drugs. You can produce. Okay, what research is going on for diabetes is now scientists are checking with removing the gene which is causing in the children. Okay, where they want to remove that gene in parents itself, because the gene is transferring from parents to baby. So they want to stop it. Okay, during pregnancy or maybe in future we will see that one, the advanced techniques and all. So why not use artificial intelligence here to create such type of receptors? The drug is available with a with every information, structure, efficacy, safety, and all, but receptor is not present on the body. So you can create a suitable receptor also. Okay. So you can alter the structure of receptor also with this genomic technology. Role in pharmacogenomics predicts drug response based on genetic makeup, helps in reducing adverse drug reactions, supports precision dosing strategies, which is very much important for us. And challenges and limitations, data privacy and ethical concerns, requirement of high quality annotated data sets, and algorithm based bias and interpretability issues, and high initial infrastructure cost. And few years back, everyone spoke about cell lines and preserving cell line and all. Now they are thinking about the cost, and now it is not that popular. Okay, so of course, again, AI based this type of pharmacogenomics technology may improve or not, we don't know, but the cost does matter here. Okay, everyone will think about the cost, and the companies also will think about the cost to establish the setup to do such type of research, etc. Right? Future perspectives, integration with the clinical decision support system, real-time genomic diagnostics, expansion into gene editing technologies. Okay, and wider acceptability in developing country. right? AI-powered genomic analysis is transforming mutation detection. It enhances precision, efficacy, and clinical relevance, holds great promise for the future of precision medicine and healthcare, right? So think about whether your genes are good or bad or what is the status of it. Okay, think and read this article which was there in Enadu on July 13, 2025, right? Very good. Right, with this slide, I conclude my presentation. Confidence and hard work is the best medicine to kill the disease called failure. It will make you a successful person, right? As a scientist, you think about this. Don't underestimate pharmacogenomic technology. Use it. I thank the organizers, especially MLR, Institute of Pharmacy. Of course, my pronunciation was wrong initially. I called College of Pharmacy. Ma'am corrected it, Institute of Pharmacy. And my special thanks to Dr. Arnaba Malik sir and Sandeep for providing me this opportunity to interact with my fellow teachers. And of course, ultimately, with patience, you attended my session. My special thanks to all the audience. And if you have any doubts, I am ready to clarify. If not now, you can send me a mail also, drchinnala@gmail.com. I'll respond to your mail. Thank you. Over to the organizers. Ma'am, hello.
Good afternoon, sir. Good afternoon, sir.
Yeah, good afternoon, ma'am. My session is completed, ma'am. If any questions are there, check it, ma'am. There is a question from Pradipti. In the next 5 years, which specific genetic mutation detection challenge, rare disease diagnosis, somatic cancer mutations, pharmacogenomics, profiling, or pathogen genomics? Do you believe AI will most decisively solve, and what is the single biggest barrier standing between correct capacity and that milestone? Obviously, ma'am, maybe AI tools may not be
Yes.
like available right now. Right now, to utilize this all pharmacogenomic technology and all, it is in your hand to prepare tools, AI tools. AI is a tool. That's it, right? We have to utilize them properly for pharmacogenomics. I may not answer for that, which like area may be focused. It depends on the researcher, right? And already I told you, for example, Dr. Reddy's, you take Dr. Anj Reddy's, her concept was to give a new medicine to the world. But the next generation, what they are saying is, I want to discover a new molecule which can cure 10 diseases at a time. And they are doing so much of good business than research. I can say that. So the same thing, if you are not able to like create tools for this and not interested, then we may not expect even AI tools in pharmacogenomics also. I hope I answered your question. Yeah, in patient counseling, you can utilize this. It is very much useful for you because, of course, so many softwares available to detect drug interactions or using adverse drug reactions and all. Okay, so as a dean of School of Pharmacy, how do you recommend structuring the genomics and AI literacy component within the PharmD or MPharm curriculum? Obviously, it should be there. And most of institutions are becoming autonomous. It is the facility given by UGC that 20% syllabus deviation, you can do it from the standard syllabus from Pharmacy Council of India. You check the new Pharmacy Council of India syllabus, it is like aligned with NEP 2020, and they gave utmost priority to artificial intelligence. Please check the syllabus once. And of course, I have not gone through each and every subject syllabus, but they gave utmost priority to artificial intelligence too. Whether it is B Farm, Farm D, or M Farm, pharmacist is pharmacist, okay, they have to provide service to the patient. And if they have excellent knowledge with them, they can do it. And first, as a pharmacist, of course, I am an academic pharmacist. Of course, we all, academic pharmacist and hospital pharmacist, industrial pharmacist, everyone is pharmacist, and pharmacist must have thorough knowledge of artificial intelligence, which can be useful in our service, pharmacy, all pharmaceutical services, or as a healthcare professional, how can you build bridge the gap between the doctor and nurse, doctor and patient? It is up to you with advanced knowledge using AI or whatever the technology. You have to work as a like connecting link between doctor and patient, nurse and patient, and as a healthcare professional of medicine, because we are the ultimate people who are discovering medicines. Okay, who are everywhere I am using the sloka Brahma Swarupamude Madhyanet Mahavishnum Sayam Sanjat Shivam Trimurticha Divakaraha. In sun, all three gods are there: the creator, the protector, the destroyer. The same way, a pharmacist is a creator, pharmacist is a protector, pharmacist is a destroyer of expired medicines. So feel proud to be a pharmacist. That's what I am saying. But when you can feel proud, if you have knowledge to explain about the drug to everyone, including the doctor. Okay, you should suggest the medicine, you should guide them for a better treatment. Of course, you can use this pharmacogenomic technology or AI for that. Thank you. Anything else?
Any more questions from the audience side? Any more questions from the audience side? You can unmute yourself and please ask.
Right. If not now, you can send me a mail also, drchinnala@gmail.com. Okay. Thank you.
Sir, we have few questions.
Ma'am, tell me.
We have few questions in the chat box, sir. Can I read it out?
Yeah. Yeah,
Pharmacy
phytoconstituents, how
graduate.
can we, wait ma'am, phytoconstituents, of course, the same method what we are doing for pharmacological evaluation for any chemical, new chemical entity. So the same method you can follow for even phytoconstituents also. But if it is an extract working on, for example, Chyawanprash, okay, what are the components? So many people studied it, but which component is improving the immunity, we can't say. The same way, so many Ayurvedic formulations are there, or your pharmacognostic formulations are there, but we can't say how it is working, on which gene it is working. And again, you have to depend on the literature only. Please, ma'am, what is the question?
Sir, pharmacy graduates are increasingly expected to counsel patients on pharmacogenomics test results. As a dean of the pharmacy school, how do you recommend structuring the genomics?
Yeah. Actually, if you have already, I have shown a picture that the patient is giving a card okay, to the pharmacist only. If pharmacist having complete idea about pharmacogenomics, what the pharmacist will do after getting that code, he will insert in the suitable instrument or laptop or computer and check the genomic picture and flaws in the genome picture and what are the drugs doctor prescribed, and he will explain them how to use and how it works also. Okay, your genome picture is like this, your CYP250 450s mutations are like this. That's why the regular medicine, if the patient is asking why lansoprazole, why not omeprazole, so you can clarify the doubt. Then the patient will get satisfaction with your services as a like counseling pharmacist. Okay, and he
Sir, we
will
have one
clarify
more
all the
question.
doubts. Yeah, tell me, ma'am.
Artificial intelligence models trained on predominantly Western genomic data sets, such as those from 1,000 genomes or TCGA, so may carry inherent population biases. So, how significant does this affect mutations detection accuracy in South Asian patients, and what steps are being taken to build India-specific genomic artificial intelligence models?
Done, ma'am. Good question. What was the dose of paracetamol previously? It was 500 mg. Okay, why it is increased to 650 mg? Okay, and one of my colleague, his name is Dr. Rajendran, SD Rajendran, who worked in Dr. DR Krishna sir lab in Karti University. His work was on population pharmacokinetic studies. So his work on digoxin dose and dose frequency, and he worked in MGM hospital, which is a 1,000-bedded hospital, and here in NIMS, and at one hospital in Chennai. And Mr. Kishore Permanla, he also worked on epilepsy drug for population pharmacokinetics again at Warangal, at NIMS, and another place. Okay, what they found is the dose frequency for whichever decided in Western population is not suitable for South Indian population. Right. Another important research which was carried out in my lab with Dr. V. Venkateshwar sir. He's Mr. Dr. Channamaneni Srinivas Rao. He worked for US FDA also. Now he is working as freelancer. He is in US now. His work was on organophosphorus poisoning, pesticide poisoning. So he worked in MGM hospital, and he published a paper on like mortality of this organophosphorus poisoning in Warangal area. And a scientist, Dr. Michael Edelston, is basically a doctor from UK. And when he was read this article, he was in Sri Lanka doing work on the same project. And he came to, he mailed to my boss, Dr. Venkateshwar sir, about this article, and he asked, I want to check your observation notebooks also. Shall I come to your lab? So sir gave permission, and he came all the way from Sri Lanka to UK, London to Bombay, Bombay to Hyderabad, Hyderabad to Warangal, and he verified each and every observation notebook of Dr. Chanman and Srinivas Rao, and insisted my guide to arrange a meeting with the doctors of MGM hospital. So the doctor asked the superintendent to arrange a meeting, and as basically he's a doctor, he gave every information about organophosphorus poisoning and all. And he told the doctors that whatever the antidotes or atropine dose and everything which given in MGM hospital is subtherapeutic level. Okay, it is not suitable as like Western population. He insisted all the doctors to increase the dose, and the doctors increased it and reduced the mortality. That's what the beauty of research. So we have done research, and we have given a benefit to the society also. So if whatever the dose frequency or whichever decided in Western population must be studied here in Indian population also. not only South, Indian population, even South Indian population also. That's why population pharmacokinetics are very much important to give you 100% treatment, not 60%. Again and again, I am saying whatever the dose frequency decided in Western people is giving 60% benefit only. So they have to think about this also. That's why now doctors are saying to manage your malaise, nothing but body aches. We have increased the dose to 650 mg. But in reality, maybe the population pharmacokinetics gave the information that 650 mg only suitable for Indian population, may not be like the 500 mg. Maybe. Okay, that may be the reason. So population pharmacokinetics will give you every information before AI and pharmacogenomics enters into the dose frequency. Is it clear? Hello? Hello, ma'am?
Yes.
Sir, any more questions from the audience?
So they are hungry, already 12.
So maybe we can conclude, sir.
Yeah.
Let
Okay.
me let me present the vote of thanks. I'm Dr. Alamaram Vaishnavi, Assistant Professor, Department of Pharmacy Practice, Mari Lakshman Reddy Institute of Pharmacy. I take this privilege to present the vote of thanks to Dr. Krishna Mohan Chinnala garu, Professor Dean, School of Pharmacy, Nalla Narasimha Reddy Education Societies Group of Institutions, Hyderabad. Sir, we are very much understood with the concepts of importance of pharmacogenomics and future for the healthcare. So personalized medicine and importance of reporting of ADRs.
Yes.
I would like to thank the management of Mari Lakshman Reddy Institute of Pharmacy, Principal Dr. Arunabha Malik sir, organizers Dr. Sandeep sir, HOD of Pharmacology Department, Dr. Balamurali Mohan sir, HOD of Pharmacy Practice, and all the participants of today's session for your attentive listening. Thank you all for the successful commencing of fourth day of the faculty development program that gave insights on artificial intelligence powered genomic analysis, revolutionizing the detection of genomic mutations. I would like to thank the management and principal sir for giving me this opportunity. Thank you, sir.
Thank you, ma'am. Thank you all. All the best. May I leave, ma'am?
Sir. The feedback link of today's session is been posted in the chat box. So please provide the feedback of today's session. And the slides of today's session will be shared in the group. Email ID is also provided. Thank you, one and all.